Acts 17, starting in verse 24. Paul is addressing the pagans at Athens. His soul had been vexed in him, the Bible says. He'd been grieved because here you have this wonderful modern society. I mean, the Greeks, even in Paul's day, you know, the Parthenon and all of the temples, it was just a marvel to behold. But at the same time, there was all this paganism. There were all these gods. There were all these false worship going on. And this bothers Paul in his spirit. And he starts talking to the Athenians. And one of the things he says to them in arguing for God and doing apologetics, this is a great apologetics passage, comes across this statue of the unknown God. And he says, that's the God that I know. And that's the God that I'm going to tell to you. And he uses this, verse 24 of Acts 17. The God who made the world and everything in it, he is the Lord of heaven and earth, and does not live in shrines made by hands. Neither is he served by human hands, as though he needed anything, since he himself gives everyone life and breath and all things. From one man he has made every nationality to live over the whole earth, and has determined their appointed lines and the boundaries of where they live. He did this so they might seek God, and perhaps they might reach out and find him, though he's not far from each one of us. For in him we live and move and exist, as even some of your own poets have said, quote, for we are also his offspring. So Paul is talking to a secular audience, a pagan crowd, people who don't have a background in Judaism or Christianity. It's not their forte. Where does he find common ground to share the faith of Christ with them? He finds it in the creation. He harkens back to the created order. He says, again, that God who made the world and everything in it. This is the God I'm talking to you about. The God where he made one man, and then from that one man, all the nations of the earth were populated. And why did he do this, Paul says? So that you may know that one God. So creation is important. The doctrine is important because it has a purpose. Part of why God makes the created order is to testify to himself so that all the nations may know that God is the true God. But in Paul's day and today, we have a lot of people who are fighting against that idea. And how do they fight against the idea of the God? They try to wipe out the idea of a creation, and they try to substitute it with an accident. But there's huge problems in this. Now, whenever I've been in discussion with evolutionists, neo-Darwinists, materialists, I've brought up certain issues. And one of the issues I've brought up is the fact that the origin of life poses huge problems to the materialist, to the naturalist in this world. If we have nothing other than the laws of the universe and the chemicals and products of nature as we see them today, how do we get such complex creatures as living beings? And usually when I bring up that argument, I always get the same response. Can you guess what it is? The response is usually something like, that's not evolution that you're talking about. That's abiogenesis. That's the creation of life. Evolution, you see, you obviously don't know what evolution is, because it's not the creation of life that I'm talking about. I'm talking about the complexity of life, the differentiation of species. From the first living organism, we get everything else. And that's usually the response I get when I talk about these issues. Now, in a strict definitional sense, yes, that's true. Abiogenesis and evolution, the Darwinian model, are two different things. But let me tell you, they both need to be accounted for, and you can't have B without A. And it reminds me of a story. What would you do if someone told you, I've got a surefire way for you to make $1 million? Maybe you'd be skeptical, but you say, well, it doesn't cost me anything to listen. Tell me, what do I need to do? Well, all you got to do first is start with $1 billion, and then you invest it in a 5% fund, and then you get your $1 million. It's completely safe. Yes, that's true, but if I had $1 billion, wouldn't need the $1 million, would I? This is what I think the evolutionists are trying to do to me. As long as we start with life, heck, we can create all kinds of species. Well, time out, man. Wait a minute. There's an old story about, and it's fictional, but I think it's about, some people say it's Winston Churchill. And his high command come to him at the beginning of World War II, and they say, you know, we have to do something, Prime Minister. The German U-boats, we can't find them, we can't see them, they're submerged, but they're just destroying our freight ships and our supply lines. They're infiltrating the English Channel. There's nothing we can do. What do we do about that? And Churchill says, well, it's easy. You just boil the channel. You boil the ocean, all the U-boats will have to surface. Then our fighters can strafe them from the air. And they said, boil the oceans? How do we do that? He goes, I don't know. I'm an idea man. The rest is an engineering detail. And most engineers really appreciate that joke because they say, yes, that's exactly what my job looks like. This is what, again, I think we're facing when we start talking about the difference between the origin of life and evolution. The National Academy of Sciences is looked upon to set most of the curriculum, the science curriculums in our schools today. And they say in their literature that positing a creator for life is incompatible with the way biology should be taught. That's the National Academy of Sciences. In their book, The Search for Life's Origin, Progress in Future Directions in Planetary Biology and Chemical Evolution, okay, that's the book that the National Academy of Sciences has published. There's one chapter entitled The Evidence for Biological Evolution. And in that chapter, it includes descriptions of abiogenesis, and it leads straight in from simple to complex transitions of life. In other words, in the book, they see it as one long continuum. Chemical evolution into biological evolution into speciation. They devote another chapter to the dismissal of any creationist perspectives. So when an evolutionist comes to me and says, you can't talk about abiogenesis, that's not the topic we're talking about, I respond with, well, how come the National Academy of Sciences says it's okay to do so? It strikes me that if they think it's okay to talk about these issues, then I'm very much within my realm to talk about a response to those issues. So, that's why I want to talk about the origin of life. The reason why evolutionists are so hesitant to talk about this is because they know it's a huge, huge problem for them. They have no answers, and they're really running scared. But before we talk about the origin of life, we have to define what the heck life is anyway. We got to be careful in our definitions because I've heard all kinds of things from mud forming and supposedly replicating through a chemical process, and that's considered alive, and things like this. You know, what is life? What do we mean when we talk about this thing that's alive? Well, the simplest definition, of course, is living is something that's different than dead, right? I mean, we don't really need to look to the dictionary when we say look at a pet and we say that pet is either alive or it's dead. We can kind of catch that. So there's a vital and functional being as opposed to one that's simply non-functioning, decaying in that respect. Webster's would maybe define it as a principle or force that's considered to underlie the distinctive quality of animate beings. A principle or force that's distinctive. In other words, even Webster's having a hard time exactly saying what life is. It's like, you know it when you see it, you know? Life, pornography. You know it when you see it. That's all I can tell you. Or dictionary.com says the condition that distinguishes organisms from inorganic objects and dead organisms. Well, thanks. That's called a, you know, it's like looking up the dictionary says, what's the word redundant? See redundant. You know, it's okay, what's life? It's things that aren't dead. Okay, you really haven't helped me there, you know. So life is a tough thing to define. It really is. But we understand that living beings, they replicate in some way, shape, or form on a cellular level as well as on a being level, at least. So there's an idea of replication and there's an idea of thriving. Now, I'm sure there's some Star Trek fans in here, but that quality of life, that essence, that vita, that's something unique. See, Star Trek's data will never be alive. The idea of a sentient or an artificial intelligence is actually an oxymoron. It can't happen. All computers do is and if anybody's ever been programming a computer, you know that one comma can ruin like a whole day. One misplaced comma will keep you checking that code. Why? Because the computers are not smart enough to know how to fix themselves. They will only do what they're told. They can't reason abstractly. They don't have a will of their own. They never will. See, the Bible would define life as a body and as a soul. There's an immaterial aspect to the being that God himself provides. Adam, right? God breathed into him the breath of life, the pneuma. That idea, that soul. Adam becomes a living being. It's body and soul together that shows life. So this is what life is. Life is something unique. It's special. It's given by God. And God has the ability to take it as well, he says in his word. But when science tries to figure out life, they only are limited to certain tool sets. So they have a couple of different ways they would do this. One would be the top-down approach. And what the top-down approach is kind of like what a lot of young kids did when they got an old radio. They say, I wonder how this thing works. And they start taking things off of it, right? Let me see how this thing works. People ask me, because I do work with computers and things like that, and people ask me, well, how did you get so good at working with computers? And I said, well, it's not that I know so much more than you. It's not that I'm all that smarter. It's just that I was the guy who wasn't afraid to say, oh, what happens if I do this? And then you see the smoke and you learn how to change a power supply. And now you've got that additional knowledge with you because you were the guy that said, I'm not afraid to do this. And then you tell somebody else, no, you don't do that. Trust me. So, and then that's all that is. So this is what the top-down approach is. You start taking things off. How low can I go? How simple can I make something until it dies? Okay, take it back one step, and now we know that that's the necessary requirements for life. The other way to do it is some people try a bottom-up approach. Let's start combining chemicals and see if something will replicate. How many chemicals do we have to combine? Things like that. Well, there's a guy, J. Craig Ventnor, and I don't know if you're familiar with him. He's a genome expert, and he works at the J. Craig Ventnor Institute with a group of scientists, and he's been working on this problem of life for, oh gosh, at least 15, 20 years now. And what Ventnor did was he was really unique, and he did it both ways. First, he took a bacteria, and he started stripping things off. He actually got it down to the smallest number of base pairs of DNA that you needed in order to keep the thing alive. And he ran all those through a computer, basically, and he captured them. Then what he did was he put that aside, and he took a yeast cell, and he took the 4 chemical components of DNA, all right, the 4 nucleotides that make up the DNA molecule, and he told the computer, put those nucleotides together, and he took that DNA and he put it back into an empty host and created synthetic life. So he started from the top down, found out what the littlest information he needed was, and then he took chemical components, put them together to construct a bacterial cell that is fully controlled by a synthetic genome. Now, if you hear about this stuff, wow, they've made life in the lab. No, it's not the same thing. An analogy I can use is maybe you have a VW, right? And you take that VW and your friend says, I want a VW as well. I say, well, I'm not going to give up mine. He goes, you don't have to. You don't have to. I'm a great machinist. And what he does is he takes your VW apart and he measures everything. And he goes to his machine shop and he uses the machinery there to roll out fenders from plain sheet metal. And he machines out an engine cast. And he does all these things. And he assembles his own VW that looks just like yours. But there's not an original part on the thing. He's built the whole thing from scratch. Now, has he created life or has he created a VW? No, he's copied what already exists, but he's done so using different starting materials. So there's a difference between saying this scientist created new life, and Ventnor would absolutely disagree with the fact that that's what he's done, versus we've created life using a synthetic starting point, using a synthetic raw materials. So that's what we see with Ventnor. Now this bacteria that he created, it's very interesting. It's called Mycoplasma mycoides. And it took 1.08 million base pairs of DNA. That's as small as he could get it, okay? So it was in the way Ventnor did it was he had 1,078 cassettes, if you will, sections of DNA, and each of those had 1,080 base pairs long, and he would combine those and he'd put kind of the whole thing together. The complete synthetic genome was isolated from the yeast cell, transplanted into recipient cells that have had the genes for its restriction enzyme removed, so they restricted it so it wouldn't replicate, and they then threw those away. And then the synthetic genome DNA was transcribed into messenger RNA, which in turn was translated into new proteins. After 2 days, viable cells which contained only synthetic DNA were clearly visible on Petri dishes containing bacterial growth medium. So Ventnor did this. And I'll tell you, this is an amazing feat. At this level, you know, at this microscopic level, this is an amazing thing for someone to do. It truly is. But it's still reading out of somebody else's playbook. It doesn't answer the question on the origin of life at all. The interesting thing, I think, and when people bring up Ventnor, I always bring this part up, is that Ventnor and his team, in order to distinguish their cells from the naturally occurring ones, because after they start replicating, if it's the same blueprint, you know, if you're really good as a machinist, and it's hard to tell whether this is the fake or the real, you know, how do you tell the difference? Well, he did some interesting things. He would put little Easter eggs in the DNA sequences that he thought were not necessary. So he put little literary passages. He encoded the names of all of the guys who worked on the project into the DNA. And he even put an email address as an Easter egg. And I guess if the first person who's transcribing the DNA found that this was an email address, they're supposed to send an email to him and they win some kind of a prize or something. I'm not sure what it was. But what does that tell you about DNA? That it's not accidental, is it? Because if you can identify an email address in DNA, then it's a messaging system. You're sending a message inside the very DNA. Huh. It sounds like there was intelligence in putting an email address in the DNA. It sounds like there was a designer who designed the email address to fit inside that DNA. Amazing. So, does Ventnor's work help us in the origin of life study, getting the life from non-life accidentally, randomly, naturally? No, not at all. Matter of fact, I think it argues for the fact that you need an intelligence. Now, whenever I talk to evolutionists, I have a little mnemonic that I use in order to talk to them about the basics of the problems of evolution. Lice. I know, people go, ooh, lice. Lice are terrible things, aren't they? I mean, they're hard to see, but when you get them, you can almost never get rid of them, right? They're everywhere, and you really, you can't, it's almost like you have to sterilize your whole house. I think evolution is like lice. I think once these guys catch it, they don't realize sometimes that they've got it, but they can never get away from the idea. It's like it always stays with them. So I want you to remember, and I'm going to go through at least the first 2 letters of my lice argument for evolution. And maybe we'll come back sometime and do the last 2. The lice argument is A, it's first, the leap to life. The L is the leap to life. And that's what I'm going to talk about tonight, the idea of the origin of life. The I is the introduction of information. So the leap to life, the introduction of information. The C is that our cells are created complex. And the E is that evolution has an edge, an end point. So the leap to life, the introduction of information, the fact that our cells are created complex, and evolution has an edge. Michael Behe is primarily working on those last 2 with a couple of his books. So I want to focus on the first 2. First of all, the leap to life. What would it take to get life from non-living material in an environment with only the laws of nature and chance? Well, the story goes that the primordial Earth was a hotbed of chemical activity. You have the idea of a primordial soup. Some people have started to dismiss that now. You have the idea of all these different kinds of, first of all, it was in tide pools where lightning would strike it. You have Stanley Miller and Urey's experiment there. And you have, then they decided that the problem with that is that it's, the early Earth's oxygen environment was actually much different than what Miller-Urey tested, so that didn't work. So they made maybe thermal heat vents under the ocean, all these kinds of propositions, but you have to have some kind of basic chemical soup, basic set of starting chemicals, kind of like Ventnor's chemical flasks that he used before he mixed them all together to make the DMA. And you have to have some kind of energy source. Lightning, ultraviolet radiation, thermal vents, something to help generate this chemical reaction so that we can get products of life. Not life yet, but products of life, such as amino acids, sugars, polypeptides, and polynucleotides like DNA and RNA. The building blocks of life. So we need chemicals, we need some kind of energy just to get the building blocks. Now, there's been some limited success in the laboratory with trying to do this. I don't think it's nearly as far as they make the claim, but there is. But it's the next step that's the big problem. The next step would be acids and nucleotides linking up and forming chains of polypeptides and polynucleotides that have the ability to self-replicate. So now instead of building blocks, we actually have something that's dividing, replicating, and coming back together. Okay, this step from number 3 to number 4, nobody can figure out how to do it. I mean, they've been working on this probably for some 70 years, and ever since, Watson and Crick figured out the DNA's double helix model, they've been trying to figure out how this would work together. And there's huge problems. Now, once you get the nucleotides replicating, then you got to put them in a container so they don't just kind of float away from each other, right? You need microspheres, like a nucleus, and blastospheres, like the cell wall itself, and then those start to replicate and divide. Now you've got an organism that's alive, multicellular organism. And then cells reproduce and natural selection favors. So you've got to go all the way to step 6 before true evolution kicks in. But it's that step 3 and 4 thing that's hugely difficult. And I'm going to give you some reasons why. Number 1 is the primordial problem. The primordial problem. I talked a little bit about the Miller-Urey experiment, and you should see a picture of it on your handout. Stanley Miller was a grad student, I believe, of Urey's, or Urey was a grad student of Miller's. I can't remember which now. I think Stanley Miller was the grad student. Anyway, they wanted to work on this idea. It was the other way around? Okay, yeah. They wanted to work on this idea of constructing these basic building blocks for life. And they started with hydrogen cyanide, water, methane, and ammonia. This is what they thought the early Earth environment was. And what he did was he sent an electrical charge through on regular intervals, and he walked away from it. In a week, they came back and they studied the results. And they had several amino acids out of these basic components that they thought the early Earth had, you know, chemicals that build the proteins at the bottom of the flask. Now, granted, it was very small. It was like 1.08%. There's a bunch of tar and there's a bunch of other stuff, but there's 1.08%, I think, of adenine, and I think guanine was in there in very small amounts. But they said, ha, we've done it. And to this day, you will see Miller-Urey's experiment referenced in most high school biology books. The problem is that, as I said, the atmosphere that we're finding now was actually rich in carbon dioxide, and maybe it even contained some oxygen. And if that's true, when you add those chemicals into the Miller-Urey experiment, the whole thing falls apart. It never produces anything. And they've been doing this for 50 years. So everybody believes that the Miller-Urey experiment is actually wrong. And every scientist holds to this, yet they don't correct the textbooks. Also, we believe that the early Earth atmosphere probably had no methane in it, or very, very little methane in it at all. And if you do that, if you pull out the methane, same result. Nothing works. So you can't even get started with the building blocks. You have these primordial problems. They haven't figured out how to get the building blocks into shape in order to form yet. But beyond that, and probably even worse than that, even if you get the building blocks, there's an organizational way that life works. There's a complex way that life works that really poses a difficulty for science. First of all, DNA. Now, if you remember your high school chemistry, DNA, you have a sugar backbone, right? Sugar phosphate backbone. And then you have these 4 chemicals that form the rungs of the DNA ladder, if you will. And they only connect in a certain way. And this is how the DNA can transfer the information, right? There's adenine, cytosine, guanine, and thymine. And then when they split apart and the RNA comes in, the difference between the RNA is it uses uracil instead of the thymine, so it makes that differentiation. But these little acids are called nucleotides, okay? They're specific little acids that bond to that sugar backbone and then line up in a certain way so that only the two will line up. Only adenine and thymine will line up together. So if you have an A here, you're going to have a T here. If you have a C here, you're going to have a G here. So it's a 4-digit code that always stays in order. Well, there's an interesting thing about all these nucleotides. It turns out that they're not as simple as some would think. The way the chemical molecule is constructed, it can be constructed in one of 2 ways. Like my hand. You see my hand? This is a normal hand, 4 fingers and a thumb. Now, you could define a hand as 4 fingers and a thumb, and that'd be fine. But there's a difference between my right hand and my left hand, isn't there? Although they both can perform the same functions, they're mirror images of each other. Well, nucleotide molecules function the same way. There are right-handed and left-handed nucleotides. This is called chirality. And when you start making nucleotides in the lab, guess what happens? You get 50% of each, because that's what randomness gives you. That makes sense. Well, here's the hook. Only right-handed nucleotides make life viable. So you have 50% right-handed, 50% left-handed, but only the right-handed ones can work for life. Now, amino acids that help build proteins, they're handed too. But guess what? It's only the left-handed amino acids that are used in building proteins. And a left-handed amino acid will cancel out a right-handed nucleotide. So you need left-handed. I mean, think about this. Imagine dealing a deck of cards, right? And you only need to deal the black ones 1.08 million times. Because if you deal a red one, you die. That's a big problem. That's a big problem. And then, but the red ones we all need. We keep those because they're the things that build the proteins that allow the rest of the cell to function. Don't mix the red cards building the proteins with the black cards building the DNA. because you'll die. Big problem. If you want to know how this works in real life, well, we had a little incident, and this is Jonathan Safarti's reference that I'm using. Any of you remember in the early 1960s, and especially in Britain, there was a bunch of doctors who were trying to help mothers who had just become pregnant overcome their morning sickness. And they gave them a drug called thalidomide in the early 60s. Remember thalidomide? Right. What happened to thalidomide babies? Deformed. They were deformed. Well, why? Thalidomide in your body will produce amino acids. It produces left-hand amino acids, which makes a very good tranquilizer. But it also produces right-hand amino acids. And that's the part that started messing with the fetus. So here we have, in actuality, a result of what we see when we talk about this left hand, right hand, in a living organism already, by the way. I mean, we're giving ourselves a big starting point. So I think that's fascinating. How do you solve that problem? That's a huge issue as far as I'm concerned. Here's another one. DNA to proteins require enzymes. In order to go from a DNA string to build a functional protein, you have to have an enzyme that unzips the DNA, that replicates it using the RNA, the mRNA, and then takes that chain and begins using other chemicals to build the protein and fold it in the proper way in order to make the protein that the cell needs. That's how this works. Enzymes tell the DNA to unzip, to replicate, to translate the strand into proteins, and then those proteins perform all the other aspects of life. But if we start with a DNA molecule, or even if we start with an RNA molecule already formed, where did the enzymes come from? How do you get life, even if you start with DNA, if you don't have the enzymes yet? Where do we get enzymes in our body? From our DNA. So now you've got a perfect chicken and the egg problem for the evolutionist. David Berlinski, in talking about this, said, this is what happens. We get a familiar figure when we start talking about enzymes and DNA. It's the figure of the chicken and the egg. First of all, he says, where's the cytosine? Cytosine, one of those 4 DNA components. He goes, that's one of the 4 molecules that make up DNA. Each one of those 4 is crucial to having the DNA message read. But according to Berlinski, quote, not a trace of the stuff has been found in any meteor, fossil, or frozen in the Antarctic. It cannot be produced by any experiment in prebiotic chemistry. Beyond the living cell, cytosine has not been found at all. So one of the 4 building blocks of DNA is nowhere to be found. We can't make it. It's not shoved under a rock someplace. There's nothing. If we can't find that nucleotide, we can't make DNA. If we can't have DNA, we can't have proteins. And if we can't have proteins, the proteins kick in the enzymes in order to cause us to make more proteins. Huge, huge problems there. So we've got the problem of chirality. First of all, we've got the problem of making the building blocks. But beyond that, we've got the problem of chirality. We've got the problem of the enzymes and the missing chicken and the egg scenario. Lastly, we've got the problem of what I call telos. Telos, this is what I say, the ends justify the mean. Life has a purpose. We have an end. We have a direction to which we go. Even if you are a secularist, you will hold, a Darwinist will hold, right? You hear Richard Dawkins talk about the selfish gene. What does he mean when he talks about the selfish? Well, the selfish gene is the gene that wants to reproduce. Wants to? How does a group of chemicals want to do anything? And really, I mean, that's a significant issue. When he starts talking about these things, he injects purpose. He injects the language of creation into a chemical reaction. It's like I said, well, gravity wanted to pull me down. I didn't actually trip. The earth just wanted me closer. You know, I tried using that on a police officer once coming down a hill. No, officer, I wasn't speeding. The earth was just pulling me closer. Didn't work. Antony Flew, who was an atheist, matter of fact, one of the foremost atheists in the last 50, 60 years. He passed away in 2010. But after, he was kind of the C.S. Lewis of atheism of his day. And he became at least a believer that there is a God because of some of these arguments. He said, quote, living matter possesses an inherent goal or end-centeredness organization that is nowhere present in the matter that preceded it. There's something different about the purpose of life, reproducing, continuing on, that doesn't exist. And radioactive decay does that. It just decays. And once it's done, it's done. But there's this idea that we keep going forward, that we keep advancing. Where does that concept even come from? And does it make sense when we're talking about chemical reactions? He says, we become hungry in order to refuel. We desire a family in order to provide successive generations. All biological systems seem to have identifiable ends, both internally and ends for other biological systems. How do we explain this? Chemical reactions happen, but there is no purpose. So the leap to life is a huge leap in my book. It's something that's going to take an awfully lot of jumping around. And I think that's significant. But when we look deeper, we have another problem than the leap to life. We have this injection of information, and I alluded to it earlier, but I'll flesh it out a little bit more here. Human beings have consistently recognized that highly specified information, like a book, the posters on the wall, believe it or not, that's highly specified information. From cave drawings to computer code, they're always the result of an intelligent mind. The identifying features of intelligence are at least these three. Number one, they're complex systems. They're not simple. What do I mean by that? Well, anybody remember Jodie Foster in the movie Contact? Did you ever see that movie? And she's working for SETI, the Search for Extraterrestrial Intelligence. She's got her headphones on, and they have these big radio telescopes pointed towards space, listening for something. Now, if Jodie Foster heard a blip once a second, continuously for 5 minutes, do you think that she would run and tell her superiors? Heck no. She'd just turn the channel and listen to something else. Wouldn't even bother to stop drinking her cup of coffee. Why? Because simple sequences like that happen in nature all the time. Simple sequences like that happen in my bathroom sink at night when you have to get up and get it. The pulsars, for example, are one way that you can generate a self-replicating, very simple, repetitive signal. So in order for this to be meaningful, it can't be just simple. It has to be a complex system. But they have to be specifically arranged to form a function. Okay? Think of, again, a computer code. If I wanted to put out a letter once a second, it's pretty easy code to write. One line says choose any letter in 1 second intervals. The second line says repeat. And guess what? That happens. 2 lines of code, you get a string. What about if I had something that said M-E, 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 M-E? How hard is that? Again, that's 3 lines of code. Pick an M, pick an E, repeat. And if I drop the 273rd sequence of ME out of the chain, and you come along and look at the chain, guess what? There's no damage done to the system at all. You'll say, I know exactly what goes in there. It's an ME. Okay? So, complex systems that are specifically arranged to form a function. But what if I wanted to say, I'm going to write a program that says John loves Mary. What do I got to do? Pick a J, pick an O, pick an H, pick an N, pick a space. Pick an L, pick an O, pick a V, pick an E, pick an S, pick a space. Pick an M, pick an A, pick an R, pick a Y. Pick a period. Stop. Right? So I've got some 17 instructions for a much shorter message than a 257-line chain of MEs. Very complex, but very specified. Those letters are important. If I drop the O from John loves Mary and I put an I got John lives Mary. And people can't understand it. Okay? If I drop the M from John loves Mary and put an H, I got John loves Harry, completely different message. So each letter is important and specific. The complex systems that are specifically arranged to perform a function, and they're highly contingent. In other words, there's nothing that forces the pattern to emerge as it did. Right? That's what complexity is. That's what we see with cave drawings. Why are these cave drawings here? Matter of fact, code breakers in World War II, this is what they listened for. Complex systems specifically arranged and highly contingent. That's what they're trying to find, because the idea of a code is you try to mask it by noise, but it has to have those three features in order to be distinguishable from noise. Right? That's what white noise is non-contingent complex information. It's just every bit of information all at once, but it's highly specified and highly contingent. Now, in order to illustrate this, I want to give you two examples, and they're on the back of your sheet. In 1976, NASA's Viking 1 spacecraft reached Mars and was taking close-up photographs of the planet. First time ever we got this close to Mars. One photo caused a huge stir, and I'm sure probably launched the National Enquirer. It seemed that there was a face carved into the Martian surface that measured over 2 miles across, and you can see the picture of the 1976 Viking 1 face on Mars. This was a big deal. If there was internet in '76, they would all be, as a matter of fact, I think people are still abuzz about this a little bit. NASA released the picture with the caption, quote, "Huge rock formation which resembles a human head formed by shadows giving the illusion of eyes, nose, and mouth." Now, scientists have discovered that the rocks that look like humans are animals before. They've seen this, right? We do this with our kids. We lay on our backs and we look at clouds and we say, what does that look like to you? Why did the NASA scientists immediately assume that the face on Mars is a natural formation? Here's another piece. In Whiteshell Provincial Park in Manitoba, Canada, there are many different stones laying about, but archaeologists are fascinated by it. Number one, one of them is the one you see in photograph number 2. Now these stones are in an arrangement where they look to resemble a man as well. But nobody says this is a huge rock formation that has the illusion of looking like a man. No, the archaeologists come out and say somebody did this. Why? What's the difference between the face on Mars and the rocks in Whiteshell Provincial Park in Canada? Isn't that fascinating? Here's the difference. The face on Mars, actually, as you get closer to it, you'll find out that it's not completely symmetric. If as they study it, they find out that it's more symmetric, then they're going to start asking questions. But as they got better resolutions and things like that, they could show that the face on Mars wasn't really a face. Or a lot of it was shadows. And you can go Google this right now, and you can see later 1990s versions of the same mound, and you can say it looks nothing like this. But the order and the specificity of these rocks in Provincial Park immediately call to mind that there's no other explanation that prehistoric people did this. We don't know who. We don't know who they were. But it's obviously a message system because they're complex, they're specifically arranged, and they're highly contingent. Non-contingent patterns. We see stones put in rows all the time. Stream beds will do that. Rushing rivers will put stones in rows. But to put them in a contingent pattern where they have angles to each other and those angles stay, that's a little different. Okay? Now, why am I bringing all this out? Well, because we can see orderly systems like crystalline structures, salt, snowflakes, or a stream that orders rocks. We can see systems that carry information are complex. Okay, even a dripping faucet that's maybe not consistently dripping, it'll never send you a Shakespeare sonnet, right? But systems that are carrying information are orderly, they're complex, and they're specific to their message. And guess what? This is exactly what DNA is. This describes DNA to a T. They're orderly, it's complex, and it's specific to its message. Because that backbone that I talked to you about before, there's nothing special about it. That's like Velcro. Any of the nucleotides can be stuck on there in any pattern whatsoever. There's nothing forcing A, C, then T, then C, or even the left-handed nucleus. There's nothing, it's all generic. So what puts them in the order in order to make an error-correcting code? Morse code uses discrete dots and dashes. The stones in Whiteshell Park make stick figures. They're too specific and too complex to happen randomly, and we know that there's a mind placing them there. Now, that's why archaeologists knew why, when they looked at Egyptian hieroglyphics or even stone cave drawings, they didn't say, wow, it's amazing how the water created those dark shadows that look like a man chasing a buffalo. I bring all of this up because what I'm saying is the SETI scientist uses this guide in order to search for extraterrestrials. Is he doing science? Absolutely. The archaeologist uses this guide in order to find ancient peoples. Is he doing science? Absolutely. So what's good for the goose should be good for the gander. If we find the exact same evidence in a DNA that we would find in, say, the first 20 prime numbers being beamed to us from a planet out in another galaxy, wouldn't you say that we should use the same assumptions about it? Right? If that's science, then what we're talking about here is science as well. Okay. Language must precede the information that carries it. This is how language works. I can say, I love you. I can say it verbally. I can write it on a postcard. I can send it via Morse code. I can use semaphore. We can inscribe it like Ventnor did into DNA. But the message comes before the message system, the communications medium. The message is separate from the medium. And with DNA, you have a message system, but the message, the information that tells how to build proteins, comes before the medium. It just uses that medium in order to communicate that message system. That means that there must be a mind behind it. There's an injection of information into DNA. Now, we know information is necessary for life. There is no diversity without reproduction, okay? If you take, I don't know, water and mix it together, all you get is more water. And you can keep doing that forever, and you'll always get more water. You need reproduction in order to get diversification. But reproduction requires information because it's two different genomes coming together and coming up with a new thing. Information is inherent in the diversity of life. And we think about this again, is there any reason why Morse code would make 3 dots equal to an S? No, they just said 3 dots will be equal to an S. That works for me. You know, and we agree upon it ahead of time. So information requires a mind to agree upon the message system, right? You ever try to read Chinese? That's pretty rough. Can't read Chinese unless somebody tells you, well, this symbol means this. Okay, got it. And then you see that symbol and you go, okay, I know what, that's the restroom. I'm good. I can go in there. Oh, you didn't tell me about men's and women's. It's that symbol next to it that's the important one, right? So symbols are random, and they only mean something if we've agreed ahead of time that they mean something. There's no trace in physics or chemistry of the control of chemical reactions by a sequence of any sort of code between them. So this is the most problem. This is one of the most difficult and challenging problems with the origin of life. Half our life-sustaining mechanism, the genetic code and its sequences, are completely arbitrary. Why would this sequence of those 4 chemicals in that certain pattern create a protein of hemoglobin? Why would it choose that as opposed to something else? We don't know. So half of our life-sustaining mechanism, the genetic code itself and its sequence, could have been a 5-digit code as opposed to 4, or a 7, or a 2, or we use binary all the time. That's arbitrary. The other half, the activity of the proteins that are assembled by it, those are necessary for life. So how can they rely on each other in some non-governed environment like primordial superchemical reactions? It's a big issue. How do you deal with the information required in order to produce a life if the medium that you're working in has no way to set what that information is? You have a coder and a decoder. If you don't have both ahead of time, then the message that you send in between the two will never be understandable. So you have the leap to life, and you have the injection of information. Okay? And like I said, the other two pieces of the lice argument is the fact that we're created complex, irreducible complexity, and the fact that evolution has an edge. There's actually not enough time to get the evolutionary diversity, even using the models that we're talking about now through the secular framework, and I just don't have time to get into that. But LICE, lice, I believe that most evolutionists are plagued with this lice argument. And if they don't solve it, they're going to be sitting around scratching their heads. That's what's going to happen. Okay. In conclusion, I think that the idea of the origin of life is a theory that can never start. One day, a group of scientists got together and decided that man had come a long way and no longer needed God. So they picked one scientist to go and tell God that they were done with him. Scientists walked up to God and said, God, pretty smart now. We've decided that we no longer need you. We're to the point where we can clone people. We can do many miraculous things. So why don't you just, go get lost? God listened very patiently and kindly. And he said, after the scientist was done talking, very well. Well, why don't we have a contest to see how well you are? Let's go make a man. Let's have a man-making contest. Scientists replied, okay, great. But God said, no, we got to do this the way I did it originally with Adam. Okay, start from the beginning. Sure, no problem. And he bent himself down and grabbed himself a handful of dirt. And God looked at him and said, no. You get your own dirt. See, if life cannot begin, then you don't have the first cell to start evolution. If science cannot posit for or against God at the origin of life, then they can't rule God out in terms of the diversity of species. If you need $1 billion in order to make 1 million, what good is the theory of making 1 million? If you can't get the billion to start with. You can't boil the oceans to make the submarines rise. Okay? Evolutionists have an origin of life problem. And the whole system has to work together as one unified theory. Any cracks there, and you'll find it breaks down. Paul was right when he used the origin of life, the creation of man, the creation of the world, to say, this is the God of whom I'm talking about. You don't know him because you're focused on all these little deities that do these little functions. I'm talking about the whole picture, the God who created the heavens and the earth. The whole thing works together as a unified theory. Unified theory, isn't that what we're looking for nowadays? Maybe in a different context, but still it works as well. You can't say this function works well, but we could care less about anything else around it. The origin of life poses a problem, and I think the lice argument is one way to show that the biblical model has the goods.